and give you guys a little bit of update on Miromatrix, what we're doing, and more specifically where we're going. So at a high level, our mission is to eliminate the organ transplant waiting list by bioengineering transplantable organs. There's tremendous need associated with this, with over 120,000 patients being on the organ transplant waiting list today. But more importantly, how do we do this? And we use this through using our patented technology called perfusion decellularization. That's our ability to actually take a whole organ, place a mild detergent through it, and wash out all the cellular material, leaving us with the perfect scaffold, then to be able to seed that with human cells and ultimately be able to create that transplantable organ. Just some quick history on us. We are a private biotechnology company. We were formed in 2009. We have very broad and extensive IP relating to perfusion decellularization and recellularization. To date, we've raised $47 million. In our last round, we brought in our large strategic Fortune 500 investor as well. Internally, our lead programs are led by the bioengineered liver. Tremendous need there. And our second product is a bioengineered kidney. How we do this, I got a nice little schematic down on the bottom that shows this. The notion is we're able to take an organ that was naturally going to be discarded from a pig, same kind of overall anatomy associated with it. We use our technology, perfusion decellularization, to remove all of that cellular material. And what that allows us to do is place that into a bioreactor. Our first product's actually coming from primary cells, from organs that don't quite meet the criteria for transplant. We can still isolate out those viable cells. That allows us to then re-engineer that organ using primary cells, which we feel has a faster regulatory cycle to it. And then the second product, once we get that in the market, we're able to solve that patient need, increase the market in terms of transplantable organs, come in with a stem cell-based product that then allows us to then re-engineer that to be patient-specific, kind of the holy grail of transplant where you no longer need immunosuppression. Now, the open question that we get, and we'll cover this a little bit probably in the question section, is that's great. How do you know that you can use a pig organ for this, and why do you not use a human scaffold? So that's something that we looked at early on, and what we decided to do from that to answer that question is actually commercialize a scaffold by itself. So we take whole porcine livers, perfusion decellularize them. We've launched 2 commercial products based off of that. So we've successfully demonstrated that we can manufacture and commercialize those. So that's really given us a leg up on de-risking the technology. More importantly, grabbing that safety profile for the matrix by itself before we put on cells. So how this looks on our clinical pathway, how are we going to get all the way to the clinic? We break this down into 4 different buckets. First, prove perfusion decellularization is effective. Just kind of talked about that with our existing products. Then how do you add cells back in? How do you recellularize this construct to get it to a point that it's transplantable? And then move into what we call a pig rescue. How do you demonstrate true functionality in a large animal model by actually removing an organ, placing our bioengineered organ back in, and demonstrating functionality before then entering human clinical studies? So just an update, again, in a limited time, what I want to show you is we're making great progress. So here's actually a liver that's been revascularized. And if you go ahead and play this video, is now being transplanted back into a pig. And what you're going to be able to see real time is the perfusion of blood through this graft as it primes up, right? So now it's actually the host, or the pig, is now circulating our construct. It's still going to be white at the top there because the circuit's not complete. Now we remove that, and now you can see complete perfusion. So now we've got that scaffold, right? So we're taking something that's already cleared as a medical device. Now we're adding cellular material back on top of it, and we've got great in vivo demonstration so far. This is an angiogram now where we take that to the next step, and now we're adding in hepatocytes as well. If you go ahead and play this video, now you can see how nicely, maybe you want to reload that a second there. Yeah, now you can see the perfusion all the way down to the capillary beds, right? So now we've got functional cells in there. We've got the vasculature, and we're able to demonstrate that complete perfusion with it. So I won't go into all this because we don't have a lot of time, but we've now initiated an acute liver failure model where we re-engineer that liver and place it back in. If we look at our controls, they go 22 to about 35 hours. Our goal is 48 hours survival, and we've seen good data with that. So I went through that kind of quick, but if you look at that bottom slide, we've had multiple animals that have extended all the way to 48 hours. So if we put this all together and start looking at how we get to the market, what we're targeting is first in human clinical studies, probably in that 2021 timeframe as we look at it today. So quick overview, really, really fast, showed you some exciting stuff on the tissue engineering side. But now we'll step over and take some individual questions.
Thanks. My name's Kay Nakai. I'm a senior healthcare analyst at Chardon Capital, where our vision is to identify companies with high potential for investment return based on potential to improve society, value to society through disruptive innovation. So Jeff, with your D-cell recellularization process, what do you think are the key biological breakthroughs you guys have accomplished?
Yeah. I mean, I think if you look at tissue engineering overall, when you look at the evolution of it in the late 90s, early 2000s, it was the notion that can you tissue engineer? Can you take a cell? Can you put it on a matrix? Can you create a tissue? And what largely the academic community proved is yes, you can. I mean, that works, right? We can grow tissue in the lab. The challenge is that it was always transparent. You could see through it. So if we got a patient who's got an MI or in liver failure, this small little, translucent type tissue isn't going to add a lot of function. So I think the biggest breakthrough, especially with our technology, is being able to recapture that vasculature. Because with that vasculature, as you saw in the video, we can now place cells in there, bring nutrients to them, but more importantly, take away waste and allow that functionality. So I think that's the biggest breakthrough with our technology, is being able to have that perfect scaffold that has the appropriate proteins, microstructure, and vasculature to be able to support the recellularization and ultimately the functionality.
So with that, why does the liver make the best target to initially go after?
Yeah. So as we looked at all the organs, there's tremendous need across any of the organs inside of the body in terms of transplant waiting list times. But the liver is kind of unique in the standpoint that there's 40,000 patients who die of end-stage liver failure every year, and the liver is the only viable therapy for them. But there's only about 7,000 transplants per year. So there's a tremendous need from that standpoint. So from a regulatory standpoint, from a patient need standpoint, we got a patient population who has no alternative therapies. So that makes it very attractive from trying to get a product out there to help these patients. The other two really points is that the liver is the one organ that can regenerate itself. So there's also the notion that once we get this construct back inside the body, we have perfusion through it, we have viable hepatocytes and liver cells in it, is it able to further enhance and grow and function? And the third one is patients only require about 20% liver function. So when we look at re-engineering this, we don't need to re-engineer a whole liver. We need to re-engineer 20% of the liver to be able to help these patients.
So in advancing your program into pigs, what are some of the key things you've learned thus far in your preclinical studies?
Yeah, I mean, our biggest hurdle preclinically has been the fact that we want to recellularize, and we have recellularized with human tissues, human cells. So we take human endothelial cells, place it back inside the graft. So one of our biggest hurdle inside of our preclinical model is that innate immune system of the pig against the human. So we're kind of going in the reverse of what xenografts have. Their challenge of taking those organs, trying to put them into humans, we're facing that same thing in a pig. But, so far we've been able to immunosuppress to the appropriate levels to demonstrate that we've got that functionality, we've got that perfusion inside of a pig.
And given that it is a xenograft for the pig, how far out do you need to be able to show either performance or just survival?
Yeah, that's a great question. So we just submitted some data on our reperfusion model of where we actually re-endothelialize liver grafts with human cells, place them back inside a pig. We do CT scan, follow that out, and look at that long-term perfusion. And what we saw is with no immunosuppression, we could get 3 to 7 days of nice continuous perfusion inside of these animal models. With a simple steroid immunosuppression, now we're able to start pushing that out to 7 to 10 days. We enhance that even further inside of our kidneys model, and now we're over 14 days. So what we're able to demonstrate in that model is that if we have a lack of perfusion, was really tied to the immune system. It was tied to the pig host cells actively going in and attacking those endothelial cells. But what we continue to learn is be more sophisticated on the immunosuppression side to further push that out. What we're really targeting, our end goal is to achieve at least 60 days of continuous perfusion and functionality inside of our preclinical models, knowing that there's going to be this caveat of trying to balance that with that immune system.
And in thinking, further out with the human liver as a target, we all know the liver has a number of different specialized cell types, hepatocyte, stellate. So in repopulating your graft, is trying to balance those out on the front end a concern for you, or will, once you've implanted the graft and it takes, that will happen on its own?
Yeah, I mean, that's some of the beauty of I think, this technology, and not only us have reported this, but academic centers who have published on this have seen this a lot too, is the fact that you can take a milieu of cells, seed it back inside of these grafts, and they naturally home. So it's not something that we actually need to rebuild that, right? There's some signaling from the matrix or innate proteins in there that are helping drive this process. And again, we just submitted this for publication. Hopefully this will come out this summer. But to drive this point home is we seed with endothelial cells that don't have any sinusoidal specific markers at the time of seeding. And one of the, if you look at the liver matrix and the construct, once you get down in the vasculature, you have sinusoidal endothelial cells. They're very specialized in terms of fenestrations and different functionality of them. We've demonstrated that once we seed in endothelial cells that don't have that phenotype, after about 14 days into our process, we start to see the upregulation of those sinusoidal markers. We've just started doing some TEM staining, and we're seeing early functionality or structures at that level as well. It's still relatively early, but if we look at the gene expression, we look at the immunofluorescence, we are seeing the matrix confer that speciality, which I think is the other reason why this technology is working so well.
And just maybe as a last question here, where are you in your program with the pigs in terms of being able to replace and prolong a survival of a replacement liver?
Yep. So we kind of broke this down into steps, as I kind of talked about in the presentation a little bit. First step is prove that you can revascularize it, because if we can't revascularize it, doesn't matter what specialized cells we put in there, they're never going to be able to support. So we achieved that. We're writing that up right now. The second step is move into an acute liver failure model. Can we prolong the survivability in an acute liver failure model? Or at a minimum, can we demonstrate that blood accumulation of ammonia and some of these other types of specialized metabolic clearance of the liver, can we demonstrate that over 48 hours? And we just wrapped up that study with very promising data, so we're getting ready to submit that for publication soon as well. What that enabled us to do, so now we've got vascularity, we can put in the hepatocytes, we demonstrate that now we've got the early functionality of it. Now what we're focused on is the orthotopic. So all of the effort right now is actually on the full liver replacement, where we take out that native liver, we put in our graft, and then look for that longer term functionality. So great progress on that front.
And just quickly, when do you think that's something you could report on?
Yep. So our targets right now is we're working out that model. We've done the first autologous or allogeneic transplants. That data is looking good. So the model is really where we need it. Those transplants are starting in the next couple of months. Our goal is by the end of the year to at least have those initial ones demonstrating functionality, knowing that we still might need some enhancement in functionality, and then early next year be able to wrap up those studies.
Okay, well, very good. That's a really exciting program you have.
All right, thanks, Kay.